This webinar is the first part of the webinars covering the topic of GI motility. The lecture also will cover GI motility, nervous control of GI smooth muscle, as well as circulation. But the first part is predominantly going to deal with general overview of the GI tract. We'll also look at the layers of the walls of the GI tract and their overall general functions, as well as looking at the general characteristics of unitary smooth muscle, which is the smooth muscle that we'll find in the GI tract, including the basic electrical activity and its control. The second part of the webinar will cover the role of the autonomic nervous system as well as the enteric nervous system on GI motility, some effects of hormones, and some different functional types of movements in the GI tract. But we're going to start is with a general overview. I love this picture because what we'll be doing in the next few weeks is we're going to be looking at general processes that are involved in the GI physiology. We'll be looking at the process of mastication, which is where you're chewing. We'll be looking at what happens as things move down the esophagus and eventually into the stomach and into the intestine. But what we're going to do first is just kind of a basic overview, looking at things that affect motility, because things have to move through the GI tract. But later we're also going to be talking about various secretions, like you see here, that are released in the GI tract, which overall, when you think GI physiology, it's all about breaking foodstuffs down into the parts where you can absorb the nutrients into the GI tract, and we're ultimately going to utilize those nutrients. Anything that we don't digest gets eliminated, and so we'll even be talking about the process of defecation. So we'll be looking at mechanical processing of food, the movements involved, the actual digestion process, the absorption of the nutrients, and also various secretions like digestive enzymes or things like mucus. That's what we'll be doing over the next few weeks. What we want to do first is do a little review that you probably covered this a little bit in Try 1, is you should have covered the general layers of the GI tract. You should have learned about mucosa and submucosa and Muscularis Externa and Serosa or Adventitia. If you don't remember, it's fine. We're going to be covering it now. So if you look at here, say a part of the GI tract, the innermost layer, which is going to be next to in contact with the food, will be the mucosa. And so I'm kind of coming over here, and so here's the mucosa right here. So from here up, we'll talk about the mucosa. So this is just representative of one particular part of the GI tract, but we're going to look at generally, no matter what part of the GI tract, what its overall functions, what type of things you'll find in that particular layer. So if you look at the mucosa is the innermost layer. It has epithelial tissue associated with it, but the type of epithelial tissue will differ depending on what part of the GI tract that you're in. So we have things like stratified squamous epithelium. Now that would be in places like the oral cavity, the pharynx, the esophagus. Because the stratified squamous, you have multiple layers. If you have a part that is gets a lot of wear and tear, you want multiple layers. Those parts of the GI tract, you're not going to be absorbing anything, so it's okay that you have multiple layers. When you have, you think small intestine, large intestine, you should be thinking more simple columnar. You have one layer. It happens to be taller than it is wider, and I'll tell you kind of the reason why it's columnar. Now, why you want it simple? Think about, this is probably where we're going to have absorption taking place. Columnar you think, well, geez, it should be as flat as possible. You can to make it squamous, but you need to have the cells be a little bit bigger because you have so many things that you have to produce. You have to produce a lot of enzymes or various proteins, and so just those organelles make up space, and so it happens to be columnar. So in this particular thing, this is representing what you would see, say, in the small intestine. You see simple columnar. So a lot of parts you see simple columnar also. Now the epithelium can be modified. You can have things like plica circularis, and the plica circularis are kind of infoldings of the mucosa. Then you also see villi here, and if we zoomed in and actually saw electron micrograph, you could probably see the brush border, which is the microvilli. Overall purpose of the plica and the villi and the microvilli is to increase surface area. So you're going to have this times when you want to have more area available for absorption. So you'll see this in the small intestine. Now mucosa can contain glands, and the glands can release things like mucus, and we'll be looking at what's the function of mucus a little bit later, but it does release mucus, and even some hormones can be released from some of the mucosal glands. So the overall functions of mucosa: protection. So you think about anywhere you have stratified squamous, it's for protection purposes. Secretion. So if you have the glands, it's going to secrete various things for the GI tract. Helps to move things down a little bit better when you have mucus. It kind of lubrication. It also does serve a protective role also. And you also have absorption. So say wherever you have simple columnar, you're going to be absorbing things across it. So it has kind of a number of different functions. The submucosa, which you see here, kind of the light purple, all of this here is the submucosa. Now before I proceed any further, you'll notice that this thing called Muscularis mucosa, that is part of the mucosa. So it's this kind of tiny little muscle layer that's associated with the mucosa, but that is part of the mucosa itself. Now underneath that Muscularis mucosa, we have the submucosa. So all of this here is the submucosa. Now if you look at it, you can start to notice the different things that are found there. So we have some glands. So these glands are exocrine glands, and they secrete buffers and enzymes into the cavity, GI tract, so they're going to serve roles in digestion, protection of the GI tract. You also have, if you'll notice, we've got some blood vessels. You also have some lymphatics, and you also can find some nerves in the submucosa. So we also have connective tissue, obviously, so we have some connective tissue, some blood vessels, nerves, lymphatic vessels. So functions, well, nourishment. So we have these blood vessels bringing in oxygenated blood and nutrients for the mucosa itself. It's involved in the digestion process. So we have these glands that are going to be releasing some enzymes that will be involved in digestion. You'll also absorb. There will be some absorption of nutrients because once it goes across the mucosa, it's got to go into the blood vessels and be carried away. So it's going to be involved in the absorption of the nutrients, carrying it away. And regulation, the nerves. So the nerves will be involved in regulating various secretory processes. And what I want to introduce you now, which is written in bold in the second page of your notes, is something called the submucosal or the Meissner's plexus. They call it the submucosal plexus because it's right within the submucosa. It's also referred to as Meissner's plexus. This is a component of what we refer to as the enteric nervous system. the GI tract has its own specialized nervous system, and the Meissner's plexus is in control of secretions. Its primary role is to control GI secretions. As you kind of notice, it's right below the submucosa where you have some glands. So it controls secretions, and it does play a little bit of a role too in controlling blood flow. Okay, so that's also what it does. Now, if you go beneath the submucosa, you have these, for most parts of the GI tract, you're going to have two layers of muscle. So if you look for Muscularis Externa, you have two layers. You have a circular layer of muscle, and you have a longitudinal layer of muscle. So the circular muscle kind of wraps around the GI tract this way, so it's going to go, it's kind of hard with this picture, wraps around, and then the longitudinal is running up and down of the GI tract. So you'll see when it's in section, it's going to look different. Well, these are going to be involved, obviously, in movement and mixing. So the contractions of this will help to move things down through the GI tract. It's also going to be involved in mixing. So when things are going down the GI tract, you're going to be releasing enzymes in various secretions. It's going to mix with the food stuff, but you want to kind of be able for all these enzymes to reach all the parts of the food. So to get proper digestion, you have to have mixing movements to allow for digestion before you move things all the way down. So you have to have proper digestion before you get everything down the GI tract. So that Muscularis Externa will be responsible for things like peristalsis, which is a movement, particular type of movement that you see in the GI tract, and also with mixing. And we'll be talking about some different types of mixing movements that you see. But you'll also notice I've put this thing called Auerbach's, or the myenteric plexus. The myenteric plexus, this one's easy to remember. Myo means muscle. The myenteric plexus is a component of the enteric nervous system that's sandwiched in between the two layers of muscle. And so it makes sense that its job is to control motility and movement, since it's right next to the muscle layers. So you do have these components of the enteric nervous system which will play a role in the regulation of the GI motility as well as secretions. Now the outermost layer of the GI tract, the name of it depends on where we are. We have something we could refer to as a Serosa or Adventitia. This layer is composed of some simple squamous epithelium and some loose connective tissue. It will have different functions depending on where we are. So we'll have a Serosa anywhere you have kind of more of a free movement. If it's not firmly attached to the wall, it's more of a Serosa. So things like small intestine is surrounded by Serosa. The large intestine is surrounded by the Serosa. The stomach. The lower part of the esophagus is. Upper part of the esophagus is lined by Adventitia, where it's more directly connected, but the lower part of the esophagus has a Serosa. So anywhere you see more of a free movement in the abdominal cavity, it'll be Serosa. That releases serous fluid, allows for lubrication so you don't have any friction. Where you have it firmly attached to other structures, like in the oral cavity, your pharynx, upper part of your esophagus, the rectum, where you don't have much movement at all, that is Adventitia. So it's more of kind of a support type role. So you have 4 layers of the GI tract, each having general functions. earliest, or the layer closest to where the food stuff is the mucosa. Big things would be more for absorption. The submucosa underlies the mucosa. That serves a role in secretions, digestion, absorption of nutrients away into the blood, control of secretions because you have some nerves in there. You have the Muscularis Externa, more about movement and mixing type events. And then you have the outermost layer, the Serosa or the Adventitia, kind of more of a support type function or allowing for free movement in the abdominal cavity. Now, in your notes, for the next few pages, you have what's called smooth muscle review. This pretty much is kind of a condensed version of the notes that you had for Physiology 1, because you listened to a webinar that covers smooth muscle as well as cardiac muscle. So I have a condensed version of those notes. You can read them on your own. It's important that you review smooth muscle, its general structure, where you find them. Obviously, we're talking about GI physiology, so we're going to have smooth muscle. And specifically, the type that we're going to be discussing is unitary. Okay, so we will be kind of mentioning that very soon again. You should have general idea of functional characteristics, what controls them, and some energetics. So you can read those on your own. Some of this stuff I'll be saying again, and in lecture I'll review with you again on this because it's important that you just you can read and just, it's pretty much self-explanatory. So I'd like you to review the next few pages in your notes that's going to review with you smooth muscle. What we're going to do is kind of maybe take it like a step further. Things that we didn't cover in Physio 1, in particularly looking at the electrical activity, because we didn't really cover that. But one thing that I did cover, but I'm going to mention again, so if you look in your notes where it says GI smooth muscle, axis, and syncytium, that's where we are in your notes. So there is unitary smooth muscle found in the GI tract. And having this particular type of smooth muscle allows it to act as a syncytium, kind of as a unit. Well, they have these gap junctions. So you see kind of where these things are kind of these arrows. The gap junctions allow the muscle cells to be coupled electrically. You'll have movement of ions through these, and so the spreading of the action potentials can occur very quickly within GI smooth muscle, so these can kind of act more as a unit instead of individual type cells. What determines the electrical activity is something that we're going to be talking about shortly, but since we're here on this picture, I could at least review a couple things on here. So if you notice what you have here, these are what we're going to refer to as not just first time I refer to it, are varicosities. So in Physio 1, you should remember that smooth muscle, the innervation is more, instead of having a neuromuscular junction per se like we did in skeletal muscle, you have these varicosities which release neurotransmitters. And the neurotransmitters you see kind of binding to certain receptors on here. It's going to ultimately, hopefully, result in the generation of action potentials and create contraction of smooth muscle. But it's a little more complicated than this. So we have to discuss a few things that we didn't discuss in Physio 1. Now, smooth muscle is way different from cardiac muscle or skeletal muscle in the fact that one of the things that you will notice, if you look here, the resting membrane potential in smooth muscle fluctuates. It doesn't stay steady. I mean, it fluctuates where it depolarizes and repolarizes, depolarizes and repolarizes. You'll see it kind of never really staying stationary. You will not see that in other types of muscle. So you have this fluctuation of the resting membrane potential. So that makes it unusual. And another thing is that the resting membrane potential can shift, where you'll still see what you're seeing here that we're going to talk about very briefly, slow waves, where these slow waves are still happening. You see these cyclical changes in the membrane potential, but they may be occurring further up here or down here. You will not see that in other types of muscle. Now the slow waves, the activity of these slow waves originates within pacemakers. So remember, smooth muscle is myogenic, like cardiac muscle. The electrical activity originates within the muscle itself. And the pacemaker cells are what we call interstitial cells of Kayes. And what the slow wave activity is, it's this cyclical change in the membrane potential. But the slow waves do not create muscle contraction. They do not. They are not action potentials. They're just the cyclical fluctuation of the resting membrane potential. So what you'll notice here, down here where he says, looks at smooth muscle tension, if you see the tension building up, we know the muscle contracted. But this did not happen until you saw the presence of these action potentials. And we're going to be talking about them very briefly. But the slow wave is just this rhythmical fluctuation in the membrane potential. It's created by cyclic opening of calcium channels. Now when that happens, calcium moves into the cell. The membrane potential will become less negative. So you see it come up. So this is it will depolarize. Okay? And then potassium channels open. Well, potassium is going to start moving out of the cell. And as it loses positive charges, you're going to come back down, becoming more negative. And so if you don't reach threshold, this little dotted line refers to threshold. If you don't reach threshold, you're just going to see this going like this. You won't see the generation of action potentials. So the slow waves do not generate contraction of the muscle. They are not action potentials. But what they will do is when you do have contractions of that particular area of the GI tract, it will determine your ultimate maximum frequency of the contractions. And it varies depending on what part of the GI tract that you're in. Certain parts, say like the stomach, the rate of the slow waves is about three times per minute. In the duodenum, it's 12 times per minute. So if we do have creation of contractions, max about contraction rate we get in the stomach is about 3 times per minute. Duodenum, about 12 times per minute. Stomach, I want you to think about why you want it to be slower in the stomach versus the duodenum. Stomach, you're kind of more just kind of mixing things up, mixing it with kind of slowly breaking up that food stuff into smaller and smaller parts, mixing with digestive juice, and then slowly it will move things into the small intestine, and you don't want to move things into the small intestine too quickly. One of the reasons why, and don't worry about writing it down now because we'll talk about it much later, is you want, when things leave the stomach, it's going to be very acidic, and you want to have time to neutralize those contents that are acidic, because you want to protect the small intestinal wall, plus the digestion of these foodstuffs do not take place very well at an acidic pH. So you want to have time to neutralize it so you can get proper digestion. So you want things to be leaving the stomach at a relatively slow rate. Now, so we have these slow waves, But you'll notice I have these little things on top of it. These are what we call spike potentials, and those spike potentials are action potentials. So the spike potentials are action potentials. They will ultimately result in contraction of the smooth muscle. So you'll see the smooth muscle tension increasing shortly after the appearance of the action potential. Now what is responsible, what you see with action potentials, is a little bit different than what we saw, say, in nerves and in cardiac muscle. The depolarization that you see in nerves, skeletal muscle, cardiac muscle was due to the opening of sodium channels. But in GI smooth muscle, it isn't. What you have was responsible for the action potentials is the opening of what we call slow calcium sodium channels. These channels are different from what you saw in nerve fibers and in ventricular muscle and skeletal muscle. These open slower and close slower than the channels that you see in the other types of muscle and in nerves. What results, though, from it is the action potential is a little long, which is good, than you see in nerves, because we want to have time for kind of sustained contraction in smooth muscle. Now, in the slow waves, now I mentioned the slow waves, this kind of going up like this depolarization was due to cyclic opening of calcium channels. But the opening of those channels is not enough calcium to be able to generate an action potential. Okay, it's not enough yet. You'll need something else in order to be able to open enough calcium channels to generate this action potential. So things like nervous stimulation or certain hormones or stretch, they will result in more calcium release that you eventually can reach threshold and generate the action potential. Now, if you look here, the picture I drew in your notes, I was trying in the blue represent those slow waves. In the yellow, I was trying to represent those spike potentials. Down here in the purple, this is the same idea where you see tension developed only after you start seeing the action potentials. What you'll notice is I've changed where these slow waves, what membrane potential those slow waves are at. So you'll notice I went from here, this first one, to this next one where I have the resting membrane potential is at a higher level. It's less negative. What type of things can cause this to happen, and what's the ultimate consequence of that? If you notice, if you depolarize the membrane potential, resting membrane potential, you're going to have a higher likelihood of creating these action potentials because you're going to be closer to threshold. And so if you depolarize, you see here and here, so if I move, this would be represented, I'm depolarizing. If you depolarize the resting membrane potential, it's going to become less negative, becomes more excitable, more likely to create an action potential, which would elicit a muscle contraction. So the types of things that can depolarize the resting membrane potential. Smooth muscle, very responsive to stretch. If you stretch that muscle, you're going to make these calcium channels leakier. You're going to be just mechanical stretch. You're going to allow more calcium to enter, or you're going to depolarize it, more likely to reach threshold and generate an action potential. The acetylcholine. And that acetylcholine could be released from the parasympathetic nervous system, so parasympathetic nervous system stimulation, as well as acetylcholine released by neurons or from the enteric nervous system. So enteric nervous system could be using acetylcholine as a neurotransmitter, as well as the autonomic nervous system, and specifically the parasympathetic branch. That will cause the membrane potential to become depolarized and increase the likelihood that you'll generate an action potential. There are certain GI hormones that also can do. So some GI hormones are stimulatory in regards to GI smooth muscle. On the other hand, things that make that resting membrane potential more negative, or we hyperpolarize, so I'm moving down here. If you hyperpolarize the resting membrane potential, you're less likely to reach threshold. You become less excitable. So things like sympathetic nervous system stimulation, which releases norepinephrine. Or it could be norepinephrine or epinephrine released by the enteric nervous system, or even the adrenal medulla. That can cause hyperpolarization of the resting membrane potential. There are also certain GI hormones that would do that. So overall, these things, if you hyperpolarize the resting membrane potential in GI smooth muscle, it's less excitable. If you depolarize the resting membrane potential in GI smooth muscle, it's more excitable. Now, to kind of change gears just slightly, but it's really not changing gears, is I want to emphasize with you the role of calcium in smooth muscle. So already you kind of noticed that I had mentioned the role of calcium in regards to action potential and also in slow wave activity. So in GI smooth muscle, the slow wave activity is because of calcium moving in through these special type of channels, unique type of channels that you have in GI smooth muscle. The action potentials in GI smooth muscle is due from the opening of these slow calcium sodium type channels. But then you also have calcium plays a role in the actual contraction of the muscle, not the stimulation of it, but the ultimate contraction of it. In skeletal muscle, all the calcium it needs to contract is in the sarcoplasmic reticulum. They have very well developed sarcoplasmic reticulum. But in smooth muscle and cardiac muscle, this sarcoplasmic reticulum is not quite as well developed. So it relies on both calcium released from this structure as well as calcium movement into the cell. So all this calcium will be involved in the regulation of smooth muscle contraction. So this right now is review. So calcium, remember, binds to a protein called calmodulin. This, when they have the interaction, calmodulin now can bind to this enzyme called myosin light chain kinase, activating it. The job of that kinase is to phosphorylate the myosin, one of the myosin chains. If that is phosphorylated, it can now interact with actin, and you can get contraction. If you have activity of a myosin phosphatase, that will remove the phosphate, and then the smooth muscle would be relaxed. Now, if you notice kind of over here, I just kind of have this receptor with an arrow here. Depending on the agonist, you have various agonists, things that can influence GI smooth muscle, regulate it. These agonists can bind particular receptors on GI smooth muscle. If they influence the release of calcium into the sarcoplasm, then that can influence the contraction of smooth muscle. So, depending on how it does it inhibit the release of calcium, or does it promote it? So various hormones can influence the movement of calcium into that cell. Things like, we'll just use general, not just GI smooth muscle. Things like vasopressin, oxytocin, those things are hormones that can influence smooth muscle. Now, the last topic I want to do for first part of the webinar is the phasic versus tonic contraction. Smooth muscle can contract phasically with very rapid contractions and relaxations, or it can contract tonically with slow, sustained contractions. Where you see these kind of phasic type ones, like as you think about things moving through the GI tract, things have to move through the esophagus, move through the first part of the stomach, the small intestine. So think about propulsive type movements. And those contraction rates can change depending on what's going on, but the maximum rate will be determined by the slow waves. So how many maximum slow waves can you have? That will determine your ultimate max rate of contraction you can get in that particular part of the GI tract. The tonic contractions. Here, tonic contractions are sustained contractions. They can maintain force for prolonged periods of time with little energy utilization. And that kind of brings me to another review of smooth muscle. Smooth muscle has this ability to maintain force for very prolonged periods of time with little energy expenditure. Here, the reason why is because of that latch state mechanism. So if you review the latch state mechanism, that is the reason why you can have that maintain force without a lot of energy, which is kind of important because certain places in the GI tract you want to kind of maintain steady contraction, not to use a lot of energy. And where you see, I have some examples of places where you'd see maintained contraction. Think about places where you want to control movement of things from point A to point B. You don't, the LES is lower esophageal sphincter. So this is going to control movement of things from the esophagus into the stomach. You want to control movement of things from the ileum into the large intestine, cecum. The internal anal sphincter, that is smooth muscle. That's going to control things going outside of your body. So you have the tonic contractions. So you have either phasic or tonic contractions in smooth muscle. What's responsible for that? Why you can have tonic in some places and phasic in others? There are differences within the myosin chains, the heavy and the light chains. You don't have to write that down. It's not important that you remember that. But you have differences in the type of myosin chains that you have in the GI tract. Now what we're going to do for the second part of the webinar is discuss nervous control. Look at the role of the enteric nervous system as well as the autonomic nervous system. But I have this animation that kind of covers a few things, a little overview or a little review for smooth muscle, but it kind of talks about the role of the acetylcholine and norepinephrine too. So I just want you to watch this animation. Come on.
Contractile mechanism can be studied using an isolated preparation of smooth muscle. Two electrodes measure the membrane potential and thus provide a record of the action potentials that result in muscle contraction. The muscle is anchored to a device that applies force to stretch the muscle. The force of the contraction is then measured on a chart recorder. Using this apparatus, researchers discovered that external stimuli were not necessary to activate the contractile mechanism. Simply stretching the muscle was enough to trigger action potentials and contraction. The cells of visceral smooth muscle are connected via gap junctions. These gap junctions provide an electrical coupling between the muscle cells that allows for a rhythmic wave of contraction to spread to adjacent muscle cells and eventually across the entire tissue. This can occur even in the absence of neural stimulation. In addition to the rhythmic self-excitation described above, many smooth muscle tissues also respond to input from the autonomic nervous system. The autonomic nervous system has two components, the sympathetic division, which uses norepinephrine as a neurotransmitter, and the parasympathetic division, which uses acetylcholine as a neurotransmitter. The isolated smooth muscle preparation can be used to study the response of intestinal smooth muscle cells to this autonomic input. Click on a pipette to apply acetylcholine or norepinephrine to the intestinal smooth muscle cells and note the response. Use the wash pipette to remove the transmitters from the preparation between applications.
Let's watch it one more time with norepinephrine. Watch here as well as here. You're seeing less force and you're seeing less generation of action potential. So the sympathetics are inhibitory. So more force being generated, increased action potential. So the parasympathetics are stimulatory. And get through.
Between applications. What happened when you applied the two transmitters? The graph shown here reveals the response patterns for the two neurotransmitters. Drag the labels for acetylcholine and norepinephrine to their correct locations on the graph.
Yay, Dr. Kayes got it right.
The results of this experiment show how the sympathetic and parasympathetic divisions of the autonomic nervous system work in opposition to each other, causing an increase or decrease in activity. In the digestive tract, norepinephrine hyperpolarizes muscle cells, which slows digestion, and acetylcholine depolarizes muscle cells, which facilitates digestion.
So let's take a quiz. Which of the following statements is false? Smooth muscle cells have only a single nucleus. Smooth muscle can contract in the absence of neural activity. The actin and myosin filaments in smooth muscle are arranged in sarcomeres, which give the muscle a striated appearance. Or all of the above statements are false. Well, smooth muscle cells do have a single nucleus. Cardiac muscles, you may have one or two per cell. Skeletal muscle is multinucleated. Smooth muscle can contract in the absence of neural activity. That's what they said at the beginning of this animation. The actin and myosin filaments in smooth muscle are arranged in sarcomeres. No, they do not have a regular arrangement. That's why they do not appear striated. So they're not in sarcomeres. Okay, next one. Smooth muscle in the gut is modulated by the autonomic nervous system, which we're going to talk about again in the second part of the webinar. The sympathetic division releases what neurotransmitter and what is its effects. So the sympathetics, you should remember, it's norepinephrine, so it's going to be one of these two. And what you just saw in the animation, it's inhibitory. Acetylcholine is excitatory. And the last question, what's the function of the gap junctions that are often found in smooth muscle tissues? Do they create a physical barrier that prevents signals from passing from cell to cell? To provide an electrical coupling between the cells that allow for a rhythmic wave of contraction to spread to adjacent muscle cells? To block the inhibitory effects of norepinephrine? Or to facilitate stretching of smooth muscle cells in visceral organs? The answer should be electrical coupling. There, we're done.